Picture this. You're in a public restroom, maybe at a university library or a slightly too hip coffee shop. You glance at the stall door, and there it is. Not a love letter or a bad joke, but a meticulously drawn diagram. The Krebs cycle in all its chemical glory. Now be honest. Did you immediately launch into a passionate explanation of pyruvate dehydrogenase? Or did you do what most of us would do, chuckle at the absurdity and move on with your life? Don't worry, this isn't a biology quiz. It's a journey into why even the most impressive feats of memorization might not matter as much as we think. For those who haven't had the pleasure, the Krebs cycle is a fundamental concept in biology. It's how our cells turn food into usable energy. Pretty important stuff, right? It's the engine that keeps us going. The biological equivalent of, well, electricity. But here's the thing. Understanding every enzyme and intermediate in the Krebs cycle doesn't necessarily make you a better doctor, a more insightful philosopher, or a whiz at fixing a flat tire. Knowing that it exists, that this elegant process fuels our very existence, that's knowledge worth holding on to. Let's face it, our brains weren't designed to remember everything. We're bombarded with information constantly, and our minds, like sensible editors, have to prioritize. Evolutionarily speaking, remembering where to find food and shelter was far more critical than, say, the structural formula of oxaloacetate. And that's okay. Forgetting is not a sign of failure, but a natural, even essential part of learning. It allows us to filter out the noise, to focus on the connections and insights that truly matter. Reading the writing on the wall, literally, back to our bathroom stall artist. What compelled them to dedicate precious time and Sharpie ink to the Krebs cycle? Were they a desperate student, cramming before an exam? Or a bored genius, leaving their mark on the world in the most unexpected way? And that misspelling, Krebs's cycle with an extra s, was it intentional? A subtle jab at the rigidity of academia? Or simply a mistake? A reminder that even the most meticulous among us are human? It's impossible to know for sure, but that's part of what makes the image so intriguing. It's a collision of the mundane and the extraordinary, a testament to the unpredictable ways knowledge can surface in our lives. Beyond the textbooks, lessons in forgetting. The point here isn't to discourage the pursuit of knowledge, far from it. It's about recognizing that true education goes beyond rote memorization. It's about cultivating curiosity, critical thinking, and the ability to connect seemingly disparate ideas. It's about understanding that the most important lessons are often learned outside the classroom, in the messy, unpredictable tapestry of life. We may forget the intricacies of the Krebs cycle, but the experiences that shape us, the relationships we forge, the ideas that spark our imagination, those are the memories that endure. Beauty of imperfect memory. So, the next time you find yourself struggling to recall a fact, a formula, or even an entire metabolic pathway, don't despair. Embrace the beauty of our imperfect memory. See it not as a limitation, but as a testament to the richness and complexity of the human experience. After all, it's not about how much we remember, but what we do with the knowledge we retain. And who knows, maybe someday you'll be the one inspiring others with a spontaneous drawing on a bathroom stall. Just maybe double-check the spelling. Unveiling the cycle of life. Krebs' pivotal discovery. The Krebs cycle, also known as the citric acid cycle or tricarboxylic acid, TCA cycle, stands as a cornerstone of cellular respiration. This intricate metabolic pathway is vital for the survival of virtually all living organisms that rely on oxygen. Its discovery, a landmark moment in biochemistry, can be attributed to the brilliant mind of Sir Hans Adolf Krebs. In the 1930s, Krebs was deeply engrossed in investigating the intricate processes of cellular respiration. His research primarily focused on the consumption of oxygen by minced pigeon breast muscle, a tissue known for its high metabolic rate. Through meticulous experimentation, Krebs observed that a specific set of organic acids, particularly citrate, played a crucial role in this oxygen consumption. Krebs's relentless efforts culminated in the unraveling of the cyclic nature of these reactions. He demonstrated that citrate was not merely consumed but rather regenerated through a series of enzymatic steps, forming a continuous loop. This groundbreaking discovery, published in 1937, earned Krebs the prestigious Nobel Prize in Physiology or Medicine in 1953. The Krebs cycle, as we understand it today, is a testament to Krebs's dedication and ingenuity. His work laid the foundation for our understanding of cellular energy production, forever changing the landscape of biochemistry. Inside the cellular powerhouse, the mitochondrial stage, the Krebs cycle takes center stage within the mitochondria, often referred to as the powerhouses of eukaryotic cells. These double-membraned organelles house the molecular machinery responsible for cellular respiration, the process by which cells convert nutrients into usable energy in the form of adenosine triphosphate, or ATP. The cycle unfolds within the mitochondrial matrix, a gel-like substance enclosed by the mitochondrion's inner membrane. This compartmentalization is crucial, as it allows for the concentration of enzymes and intermediates necessary for the cycle to proceed efficiently. The primary role of the Krebs cycle is to oxidize acetyl-CoA, a molecule derived from the breakdown of carbohydrates, fats, and proteins. This oxidation process releases energy in a controlled manner, which is then harnessed to generate ATP through a series of subsequent reactions known as oxidative phosphorylation. 8 steps of energy liberation, a detailed walkthrough. The Krebs cycle is a finely tuned metabolic dance consisting of 8 distinct steps, each catalyzed by a specific enzyme. These steps can be broadly categorized into 2 phases: the introduction and oxidation of a 2-carbon unit, and the regeneration of the starting molecule to continue the cycle. First, citrate formation. The cycle commences with the condensation of acetyl-CoA, which has 2 carbons, with oxaloacetate, which has 4 carbons, to form citrate, which has 6 carbons. This reaction is catalyzed by citrate synthase. Second, isomerization. Citrate undergoes rearrangement to form isocitrate, its isomer, facilitated by the enzyme aconitis. Third, oxidative decarboxylation. Isocitrate is oxidized, releasing carbon dioxide and forming alpha-ketoglutarate, which has 5 carbons, with the reduction of NAD+ to NADH. This step is catalyzed by isocitrate dehydrogenase. Fourth, oxidative decarboxylation. Again, alpha-ketoglutarate undergoes another round of oxidation, producing succinyl-CoA, which has 4 carbons, with the release of carbon dioxide and reduction of NAD+ to NADH. This reaction is catalyzed by the alpha-ketoglutarate dehydrogenase complex. Fifth, substrate-level phosphorylation. Succinyl-CoA is converted to succinate, generating GTP, which is equivalent to ATP, through substrate-level phosphorylation. This step is catalyzed by succinyl-CoA synthetase. Sixth, oxidation. Succinate is oxidized to fumarate with the reduction of FAD to FADH2. This reaction is catalyzed by succinate dehydrogenase. Seventh, hydration. Fumarate is hydrated to form malate in a reaction catalyzed by fumarase. Eighth, oxidation and regeneration. Malate is oxidized to oxaloacetate, the starting molecule of the cycle, with the reduction of NAD+ to NADH. This final step is catalyzed by malate dehydrogenase. Section 4. The grand finale, summarizing the cycle's output. After completing one full turn of the Krebs cycle, one molecule of acetyl-CoA is completely oxidized to 2 molecules of carbon dioxide. This oxidation process generates 3 molecules of NADH, 1 molecule of FAD2, and 1 molecule of GTP, which is equivalent to ATP. These reduced electron carriers, NADH and FAD2, play a crucial role in oxidative phosphorylation, the final stage of cellular respiration, where the majority of ATP is generated. Section 5. A universal symphony of energy, significance, and modern implications. The Krebs cycle is not just an isolated metabolic pathway, it's a central hub connecting various metabolic pathways in the cell. Its significance extends beyond energy production, playing vital roles in biosynthesis, cellular redox balance, and cellular signaling. In biosynthesis, intermediates of the cycle serve as precursors for the synthesis of essential molecules such as amino acids, fatty acids, and heme. When it comes to cellular redox balance, the cycle contributes by regulating the levels of NAD+ and NADH, and in cellular signaling, intermediates of the cycle can act as signaling molecules, influencing various cellular processes. Modern research continues to unravel the complexities of the Krebs cycle, particularly its role in diseases like cancer and mitochondrial disorders. Understanding how the cycle is dysregulated in these conditions could pave the way for novel therapeutic interventions. The universality of the Krebs cycle highlights its fundamental importance to life as we know it. From the smallest bacterium to the largest mammal, this elegant metabolic pathway stands as a testament to the intricate and interconnected nature of biological systems.
Have you ever stopped to think about what keeps you going day after day? What is it that allows you to wake up in the morning, move your body, and tackle everything life throws your way? It's something we often take for granted, but it's absolutely essential to every moment of our lives. What fuels your every move, every thought, every breath? The answer is simple, yet profound. It's energy. Whether you're sprinting down a track, solving a tricky puzzle, or just taking a deep breath, energy is at the heart of it all. Just like a car needs gasoline to drive, our bodies need a constant supply of energy to function. We get this energy from the food we eat, the air we breathe, and even the water we drink. Every bite, every sip, every breath is part of the process that keeps us going strong. But have you ever wondered where this energy actually comes from? How does your body turn a sandwich or a piece of fruit into the power you need to run, think, and play? It's a fascinating question that scientists have explored for generations. The answer lies deep inside you, in a remarkable process happening within every single cell of your body. The Krebs Cycle. This process is like a hidden engine working tirelessly to keep you energized. The Krebs Cycle is a fundamental process of life, a tiny but mighty engine that powers everything we do. It takes the nutrients from our food and transforms them into usable energy, fueling every heartbeat, every thought, and every step. So get ready to journey into the amazing world inside your cells. We're about to take a closer look at the microscopic machinery that makes life possible. Together, we'll uncover the secrets of the Krebs Cycle and discover how this incredible process keeps us alive, active, and full of energy every single day. Imagine being a scientist in the 1930s, trying to solve one of life's biggest mysteries. How do living things get their energy? That's exactly what Sir Hans Krebs did. Working tirelessly in his lab at the University of Sheffield, Krebs pieced together the intricate steps of this cellular process. His groundbreaking discovery of the citric acid cycle, later renamed the Krebs cycle in his honor, earned him the Nobel Prize in Physiology or Medicine in 1953. Krebs' work revolutionized our understanding of how cells convert food into usable energy. His legacy lives on in every biology textbook and every research lab studying cellular metabolism. Let's take a journey deep inside one of your cells. Our destination? The mitochondria. Often referred to as the powerhouses of the cell, mitochondria are tiny organelles responsible for producing energy. Within the mitochondria lies a special compartment called the mitochondrial matrix. This is where the magic of the Krebs cycle unfolds. Like a bustling factory floor, the matrix contains all the necessary machinery and ingredients for this vital energy-producing process. Imagine a water wheel continuously turning, using the flow of water to generate power. The Krebs cycle operates in a similar way. It's a circular series of chemical reactions that extract energy from food molecules. The cycle starts with a molecule called acetyl-CoA, which is derived from the breakdown of carbohydrates, fats, and proteins we eat. Through a series of steps, this molecule is transformed and broken down, releasing energy along the way. Think of it like a relay race, where each runner or molecule passes a baton or energy to the next, ultimately generating energy for the cell. Now let's break down the Krebs cycle into its 8 individual reactions. This cycle, also known as the citric acid cycle, is at the very heart of how your body turns food into usable energy. Each step is like a carefully choreographed move in a dance, ensuring that your cells get the fuel they need to keep you alive and active. First up, citrate formation. This is where the magic begins. Acetyl-CoA, which is derived from the breakdown of carbohydrates, fats, and proteins, joins with a molecule called oxaloacetate. Together, they form citrate, a 6-carbon molecule that sets the stage for everything that follows. This initial combination is crucial, as it's the entry point for all the energy stored in your food to enter the cycle. Imagine 2 cars merging onto a highway, ready for a long drive. Just like those cars, acetyl-CoA and oxaloacetate come together smoothly, setting off on a journey that will power your body's every move. Next, isomerization to isocitrate. Citrate undergoes a subtle but important rearrangement, transforming into its isomer, isocitrate. This step might seem minor, but it's essential for the cycle to continue. It's kind of like rearranging furniture in a room to make space for more. You're not adding or removing anything, just shifting things around to make the next steps possible. Then comes oxidative decarboxylation to alpha-ketoglutarate. Here, isocitrate loses a carbon dioxide molecule and is oxidized, forming alpha-ketoglutarate. This is one of the steps where energy is actually released and carbon dioxide, a waste product, is produced. This is like taking a detour on a road trip and, releasing some exhaust fumes. The journey continues, but you've left something behind, just as your body releases CO2 with every breath. After that, we have succinyl-CoA formation. Alpha-ketoglutarate undergoes another oxidative decarboxylation, producing succinyl-CoA and releasing more energy. This step is another checkpoint where the cycle harvests energy from the original fuel molecule. Think of this as reaching a scenic overlook and taking a break, rejuvenated and ready for the rest of the journey. You've made progress and now you're recharged for what's ahead. Next, succinate generation. Succinyl-CoA is converted to succinate, and in the process, a molecule called GTP is produced. GTP is very similar to ATP, which you might know as the cell's main energy currency. This is one of the rare moments in the cycle where energy is directly captured in a form the cell can use right away. This step is like exchanging your tired horse for a fresh one, ready to gallop ahead. The journey continues with renewed energy, thanks to the new fuel you've just picked up. Then, fumarate formation. Succinate is oxidized to fumarate. This oxidation step is subtle but important as it helps set up the final reactions of the cycle. It's like crossing a bridge, marking progress on your journey. Each step brings you closer to completing the cycle and starting anew. Afterward, malate formation. Fumarate is hydrated, meaning a water molecule is added, forming malate. This hydration is a simple but vital step, preparing the molecule for the final transformation. Imagine stopping for a refreshing drink of water at a roadside spring. That added water is just what's needed to keep the journey going. And finally, regeneration of oxaloacetate. In the last step, malate is oxidized, regenerating oxaloacetate, the very molecule that started the cycle. This regeneration is crucial because without oxaloacetate, the cycle couldn't continue and your cells wouldn't be able to keep producing energy. You've completed the loop and are ready to pick up another acetyl-CoA molecule, starting the cycle anew. This elegant series of reactions happens countless times every second in your body, powering everything from your thoughts to your movements. The Krebs cycle is truly the engine room of life, quietly fueling you with every breath and every bite you take. After this whirlwind tour of the Krebs cycle, you might be wondering, what's the big deal? Well, the cycle produces crucial molecules that power our cells. NADH and FAD2. These molecules are like energy shuttles carrying electrons to another process called the electron transport chain, where even more ATP is generated. ATP and GTP are the cell's primary energy currencies, powering various cellular processes. You can think of them as tiny batteries that fuel everything from muscle contractions to brain activity. And of course there's CO2, which is released as we exhale, showing the intimate connection between our breath and cellular respiration. The Krebs cycle is essential for all living organisms that rely on oxygen to survive. It's the central hub of cellular metabolism, connecting the breakdown of food molecules to energy production. Think of your muscles working hard during a workout. The Krebs cycle is working tirelessly within your muscle cells, providing the ATP needed for those powerful contractions. Similarly, your brain, which constantly processes information, relies heavily on the ATP generated through the Krebs cycle. The cycle is also linked to various diseases. For instance, cancer cells often exhibit a modified Krebs cycle, known as the Warburg effect, which contributes to their rapid growth. Understanding the Krebs cycle has profound implications for medicine and human health. Researchers are constantly studying this vital pathway, hoping to develop new treatments for a range of diseases. For example, mutations in genes encoding enzymes involved in the Krebs cycle can lead to mitochondrial diseases, which affect energy production and can cause a variety of symptoms. Additionally, research suggests that targeting the Krebs cycle could hold promise for treating cancer and metabolic disorders. From the lab bench to the clinic, the Krebs cycle continues to fascinate and inspire scientists, offering hope for improving human health. The Krebs cycle is a testament to the elegant and intricate workings of life. Deep within every living cell, this cycle operates with astonishing precision, orchestrating a series of chemical reactions that transform simple nutrients into the energy that powers our bodies. It is a process so fundamental that without it, life as we know it would simply not exist. This microscopic process, occurring billions of times every second within our bodies, is a powerful reminder that we are all connected by the fundamental principles of biology. Every heartbeat, every breath, every thought is made possible by the relentless work of these tiny molecular machines, tirelessly converting food into fuel. The Krebs cycle doesn't just sustain us, it links us to every living thing on Earth, from the smallest microbe to the tallest tree. It is a universal language written in the code of chemistry, shared by all forms of life. As the late scientist and science communicator Carl Sagan once said, "We are made of star stuff." And indeed, the atoms that make up our bodies, the molecules that participate in the Krebs cycle, were all forged in the fiery hearts of stars billions of years ago. The very elements that flow through our veins were once part of ancient cosmic furnaces scattered across the universe before finding their way into us. So, the next time you feel your heart beating, your lungs expanding, or your brain firing on all cylinders, remember that these everyday miracles are powered by a cycle that has been running uninterrupted for billions of years. Just remember the tiny but mighty Krebs cycle, quietly at work in every cell, every moment, sustaining the spark of life, the circle that powers life. It is a never-ending dance of molecules, a cycle that connects us to the stars, to each other, and to the very essence of existence itself.